We're thinking about the human microbiome and ways in which they are setting up the sampling protocol, and then what do they do with the samples, and which methods are maybe a little bit better than others. So this list is looking, this is the vast majority of papers, at least that I've read. You have these types of experimental methods going on. And probably the last bullet point, longitudinal studies, there's not very many of those completed, unfortunately, because that's really kind of the best situation if you can track a person over time. And the future of personalized medicine, that's what they hope to get to, so that you can, as a baby, your parents will bring you to the doctor and they'll start taking samples at that point, and then just working through your life, you'll be getting assessed throughout your life so that they have that data for you and can create some personalized medicine choices for you. And so that's kind of the holy grail, so to speak, of how to do this. But there's some nice longitudinal studies in progress right now, some big ones that are impressive, but they're in progress. They haven't finished the data analysis and even collecting the data yet. So with all of the ideas here, keep in mind that a lot of the papers that are published, especially some of the earlier papers, they can talk about correlation, but it's really hard sometimes to prove, to really say this microbe is doing this. It's hard. And so remember, as you've learned in all your science classes, just because there's some correlation with, this certain microbe, and all of a sudden, oh, look at, type 2 diabetes, that you have to be careful on how you come up with conclusions on that, because just because you see a correlation doesn't mean causation. The vast majority of papers tend to zoom in on the colon, which is the last part of your intestinal tract. That is where there'll be some nutrient in that spot where the nutrients will go into your blood vessels and get carried to your body. But the vast majority of what's moving through the colon is going to get defecated out. And that's where we see the largest population of microbes. So they're very interested in that. Plus, when you take fecal samples, it's pretty representative, they think, of what's happening in the colon. Not so much maybe with the small intestine, because it's hard to say when you are examining feces, like, you can't tell exactly where those microbes were in the intestinal tract. So that is a problem that has been plaguing this field of research. Like, how do we know where they were? And so if you want to do that on humans, it's not really doable, because you have to do surgery at this time in order to collect samples from the small intestine, let's say. And that's just not something people are going to sign up for, to be routinely sampled like that over time. So with mice, they can do that, but with humans, not so much. But I thought so Jessica was a former student here. She did her undergrad here and her master's with me. And then she went up to Michigan Tech University and completed her PhD up there. And her PhD dissertation was actually looking at this idea of, can we see any differences between the small intestine and the colon in terms of the microbial communities there? And so her hypothesis was that they will significantly differ compared to, well, in this case, she was also looking at the mucosa. So she did small intestine, large intestine, and then she was looking at what microbes are hanging out in the mucosa, the mucus, and then what ones were just from fecal samples. So in order to get at this question, she had to do invasive surgery, because she was directly sampling mucus, and you can't do that from fecal samples. It's too imprecise. So obviously she worked with mouse model, and was doing also, she was weaving into this what happens if you have people, or the mice in this case, exercising versus not exercising. Is there any impact on the microbiome with exercise? And she did think that there would be. So that was her hypothesis. So she spent years working through this and collecting data. I'm not going to go through all of it. I'm just going to give you the conclusions, but she did see significant, statistically significant differences between who was living in the mucus area and who was collected from the fecal samples. And so her point is, if you're looking and trying to understand who's in the mucus, fecal samples may not be the best way of sampling. So this is just an example of, you really have to think about your question and then think about how do we sample to really get at that question. And so, you know, with this idea, surgery and sampling through surgery works, but like I said, most people aren't going to do that. As far as the exercise, it moderately altered the community in the colon, but not in the small intestine. She didn't see any differences in who's showing up in the small intestine with exercise, but she did in the colon, if you're interested. And then some of what she was saying, what, you know, next steps for this, you know, what we're going to do next, said, yeah, you know, you need to know which microbes are in the mucus of healthy versus, you know, not so healthy individuals. So that longitudinal study would be great for that if we can sample the mucus for microbes. And so better observation techniques. I'll talk about the indigestible pill in a minute here. But basically looking at this idea of, you know, better understanding the communities in the different parts of the intestine, especially most scientists are very interested in the microbes that hang out in the mucus layer, because those are the more persistent microbes that stay with you long term, and they have a very close physical association with your epithelial cells and your immune system cells. And so they're very interested in those. So she mentioned this indigestible pill. So they're actually working on this. This was a podcast. I didn't get to listen to all of it, but I think it's the notes section. I have the link for it. Bugging our bowels, engineering bacteria to eavesdrop on gut health. So they're actually working in the field of synthetic biology to try to get this to happen. And so it's worth listening to if you ever have time, but basically looking at ways to engineer microbes, smart probiotics, to better design, you know, the microbes that hang out with you, to be able to see what's going on there. So this indigestible pill or lab on a pill is actually in the works right now. And so within your lifetime, I think it's going to be something that you can ask for at your doctor's and have this done. And so this is Tufts University. They were one of the leading groups that did this work, but basically they have a 3D printed indigestible pill that samples bacteria as it moves through your intestinal tract. And then they can actually tell where it sampled from and, you know, correlate the microbes to their actual physical environment. So it's like the size of a, you know, a vitamin pill or something like that. And you swallow it, and then it takes samples as it moves through. And I was looking for updates on that, and they said, yep, you know, we've made significant progress about the size of a vitamin pill that can be swallowed and pass through the GI tract to identify the full inventory of microbes in an individual. So they were kind of pioneering this idea. And right now it is awaiting human clinical trials. What do you think? Would you sign up for that? Just have to swallow a pill. And yeah, they can poop it out. They don't have to go in and extract it from you. Very good question. I'm like, how the heck are they doing that? Because so I won't go into it. You can read up on this on your own. I won't ask about it, you know, on the exam or anything, but these are different. So it wasn't just Tufts now, like now there's other entities that are getting into this type of ability to author something and make money off of it, of course. So you have UCLA getting into it and some private companies, University of Pittsburgh. But like this one, the Tufts one, polyacrylate beads to collect bacterial samples and close off the inlets, a magnet. I think what they'll do is like have you swallow it and then they'll have a magnet on you and they'll move it through you that way, I think. A magnet in the pill allows for targeted sampling, so like they can probably track or know where it is and then they'll like guide it through and have it take samples. This one releases a gel that collects bacteria, travels through the bubble and closes when pressure changes. I don't know exactly how they're gonna do that. Pulls fluid into a bladder that will seal when full. So yeah, really creative ideas here. We have a lot of different scientists and engineers, I'm sure, working on this effort. This is really interesting. So anyways, this could be something that you'll be able to do in the not so distant future. So I would do it if I was offered that. I think that'd be really interesting. Before I forget, I forgot last time, I want to ask you a Google Forms question. So let me switch over. All right. Let's see if it should be one question that I'm asking you once you get in there with several options to pick from. Is that what you're seeing? One question on fiber? Okay. Take your best guess. Oh, not everyone has taken them, but it's taking so long for that to happen. I think I'm just going to release them later today, the multiple choice especially, and probably not the essay yet, but the multiple choice, because it's just taking too long. But it's if I'm remembering, it was a very good average, by the way. So oh yes, oh yeah, yeah. Please be, yeah, I definitely will do that. I mean, I literally go through every single question on every single exam and look at the stats. So I just haven't had, I want to do that when I am not interrupted with other duties and stuff, but it will get done. And I'll announce that. Are we all answered now? Let me just see if I can see. So the number of responses goes down as the list. So a lot of you answered 10, or where's my 0? Isn't there a 0 one? 0 to 10? Was there one like that? Oh, less than 10. A couple of you answered that. Yeah, I'm gonna maybe not today, but I will talk about fiber types because that's a big umbrella. There's a lot of different fiber out there. So this is, yeah, I mean, depends on how you slice it, right? How you're going to answer this, but okay. So a lot of you were saying 15 to 20, 20 to 25. Does anyone know what you're supposed to be getting according to the people who say you're supposed to be getting stuff in your diet? I can't remember who that is. Does anyone know? It used to be 20 grams. Now they've upped it to 25, I believe. Although people in the microbiome sciences say 30 is better. And then some people are saying 50
or more. Yes. You can feel not so good from it. Like so if you're not used to taking fiber and then all of a sudden, like let's say someone takes 10 grams per day and then they're like, I'm gonna go to 60. And then they just go wham, 60. Well, good luck. You're gonna be having a lot of intestinal pain. Whether or not it won't make you get hospitalized, but you'll feel bad. You have to gradually up your dietary intake. Otherwise, and then there's special cases like people with IBS or Crohn's or all sorts of colitis. You have to speak to your doctor because they will tell you if that's safe to do. So if you have certain issues like that, you definitely need to talk to your doctor. Okay, so we'll talk more about fiber because geez, all the papers I'm reading, it's amazing how they keep zooming in on fiber, much more so in the recent literature than what they did in the past. Yes.
CMOS ads on Instagram and saying like I have to detox my gut and it's gonna give me all these benefits. So like, is that kind of invalid or?
Um, I don't know a whole lot about that. Somebody else was asking me about that in my other class too. So it must be a thing going on right now that is with that Instagram you said. So when they say sea moss, are they talking about, do they give a species name?
No, it's like CMOS gel, also CMOS capsule.
What I'm guessing is like when I hear sea moss, I think about Chondrus crispus, which is a red algae, and it does produce algin, which is kind of like pectin. So it is like a fiber source. Yeah, I mean, if that's what they're talking about, but often they won't say like what species they're thinking of. And so some of these nicknames, like I don't know exactly what they're thinking about.
Yeah.
So just, like go to their website and figure out about. So, all right, why am I screen? I don't know why it's doing that. Let me just do this again. So there's different kinds of fibers for sure. Fiber, it's a big umbrella term, and I'll get into that. But before we do that, I'll also be getting into probiotics, prebiotics, and that kind of stuff. So really interesting aspect of microbiome research is looking at the idea that the microbiome can influence other parts of your body besides the gut. And one of the most probably surprising implications of this is how it can affect your brain and your thoughts, your mood, your emotions, even anxiety and depression, and others on top of that. So the gut, your gut is the biggest digestive organ, of course, but it's also the biggest immune organ when you add up all the immune system cells in that area, an endocrine organ as well. And it possesses an enteric nervous system, its own nervous system. So your gut and the microbiota there, all the microbes, they work together to help you digest, but they're also working together for your proper immune functions, endocrine functions, and neurotransmission. So some scientists are calling it the gut brain, that whole area of your body, the gut brain, because unlike other peripheral organs, it can work without instructions from the brain. So your gut keeps going, even if your brain isn't working, so to speak. So it completes its local functions, but also regulates human behavior and cognition, similar to the brain. So when they started piecing this together a while ago, it was, like I said, a big surprise. This was not anticipated. And so this is a very active area of research currently. So here is a visual trying to get you oriented to what's going on here.
And I am not a neuroscientist, all right, so this is coming from a microbiome perspective. I'm not gonna go into all of this. I don't expect you to know the immune system and the neuroscience of this. But some key things here that they're trying to show is that you have this gut-brain axis, where they're saying you can get microbes in the gut influencing what's going on in your brain. Okay, that's a big idea from this. They're showing this lower image here, that's your gut and your epithelial cells. And here in this particular case, they're showing what happens if you have an altered microbiome composition. So they're trying to show that. And I think the one on the left, they're trying to show nice diversity, high populations, which, by the way, they're finding, I mean, the general consensus seems to be the higher the amounts of microbes and the greater diversity leads to healthy gut action and immune function and all of that. And so if you have low diversity because of antibiotics, maybe that you took or something like that, that's not ideal. As not only does it impact diversity, but also population sizes. It's pretty much kind of a universal thing. I think if you've taken BI 210, you're probably learning that with ecosystems that, you know, the greater the diversity in an ecosystem, generally it's a healthier ecosystem. I have the same idea when you get down into the ecosystem of your body and the microbes. So they're trying to show that what's going on in your gut is influencing what's happening in your brain through a variety of mechanisms. So they're showing there's the nerve action going on, but they're also showing molecules being produced by the microbes can travel. They don't just stay in the gut, they can travel and potentially all the way to the brain. Immune system cells that are influenced by your microbes can also be part of this. And so you have kind of a mixture of things that are going on that they think is allowing for this bidirectional communication. And the fact that the microbes can influence your brain, microbes in your gut can influence your brain was an amazing discovery. So here this is taken from a paper saying, well, you know, one of the issues you probably know about the blood-brain barrier, that, you know, there's very limited things that can pass that and get into your brain. And so that was like, well, how is it affecting your brain if you have a definite blood-brain barrier? So this paper was kind of looking at that idea, and they said, well, okay, if you have, like, they know microbes in the gut can produce neurotransmitters, and neurotransmitters functioning in the brain to relay messages, you know, between the synapses. And so they're like, but if they make a full-fledged active neurotransmitter, it's usually too big to fit across the blood-brain barrier. So they're like, well, that's why they're showing this is not really working. So then they said, well, it could happen where the vagus nerve is triggered by the actions of the microbes in the gut, and through the nerve function, you can get the signal being sent to the brain. That's certainly doable. And then they said, what about precursors to neurotransmitters, where they know microbes do that too? They make a bunch of different precursors that can end up being parts of the active neurotransmitters. And they said, what if those can make it through? They're small enough to make it through the blood-brain barrier. So they're really looking at this, like, how is this really working mechanistically? Because you do have a protection with your brain with that blood-brain barrier. So as they're, you know, trying to figure out all of that, they've done some studies where a lot of this is mouse studies now, okay? Although it's transferring into human clinical studies now too. But initially they worked with mice, and they were showing through mouse studies that cognitive functions, learning and memory were influenced by what was happening in the mouse gut. So antibiotic usage, they would see some differences before and after antibiotic usage, where they were looking at spatial memory and what happens if you add back some microbes. You know, is there a shift in that at all?
I have a question about this slide before with neurotoxins. Is it only neurotoxins produced by bacteria that can't get through the blood-brain barrier?
You mean the neurotransmitters?
Yeah.
Yeah, they're saying if it's a full-on active neurotransmitter like GABA or something like that. Like microbes can do that. They can make it, but it doesn't, it seems too big to make it through. They're hypothesizing that when it's fully functioning, it's not making it through the blood-brain barrier. But precursors to GABA or other neurotransmitters, they can get through. That's what they're trying to show in this diagram.
I'll talk a little bit more about that. I think I have it in a subsequent slide here. Mood and emotion, they're saying, is affected by the gut microbes. And when they start playing around with probiotics or prebiotics or even fermented food, they could see some differences in behavior, in emotions, and memory. So now they're saying, you know, we need to think about the microbiome for some cases of mental disorders, and that it could be a dysbiosis in the microbiome of the gut that's actually could be causing or definitely influencing that path into a mental disorder. So they're looking at this because, of course, if this is true, then they're thinking that, well, if we could understand that and then figure out a way to influence the microbiome population, maybe by providing the patient with a probiotic or a prebiotic, that could be way safer and cheaper than some of the drugs that are out there currently with some side effects. I'm sure you've heard like radio or TV ads for certain drugs, and the person starts talking really fast toward the end about all the side effects. And it's just kind of scary. So they're saying, you know, if we could understand this better, we could maybe approach it with a less serious issue.
I guess I've just heard conflicting takes on probiotics,
Yeah.
like especially when they first started talking about them, it was really popular. Like, how does the bacteria survive the stomach and then get cultured?
Exactly, right? That's a very good question, because what, pH 2 or so in the stomach? And so I'll talk more about probiotics, but you really got to do your research before you invest. And not only, well, and you know, really do your research on a company to verify that they're legit and truly designed a good probiotic, but they're not cheap. You know, some of the better ones out there, they're not cheap. So if you're going to spend money, you gotta do some. But you're right. And so a lot of the probiotics out there right now, if you look on the ingredient list, a lot of them are bacillus species. Do you remember what bacillus are so good at doing if they have to? Endospores. Endospores can survive your stomach, no problem. And so that they've really latched onto that idea. And you'll look on the probiotic list and they'll usually say like Bacillus coagulans, and then they'll put in parentheses, G55. It's their proprietary strain that they've used. So look for that. So bacillus species are endospore formers, but there's a lot on like the probiotics I take sometimes. There's a bunch of other ones there too. But then they design the pill to be slow releasing, I think they call it, or something like that. So it makes it through your stomach intact. But that's another matter. Yes, the endospore, it'll go through your stomach, no problem. And then it'll set up camp in your intestines and germinate and grow. Yep. And so, or if it's a slow releasing gel capsule or whatever that can survive your stomach, like the one I take, they have that documented that it can survive through the stomach intact. So that's another one. They're, looking at really serious issues here like Alzheimer's and Parkinson's and things like that too. And surprisingly, they are finding, I already mentioned to you a while ago that they found microbes from the mouth can get to the brain, and it seems to be associated with Alzheimer's, but now they're seeing gut microbes too are influencing the Alzheimer progression. And so they're looking at this much more carefully. I'm sure all of you are aware that this is such a so many people have Alzheimer's, and it's just getting worse and worse. So this is really a serious issue that needs lots of different points of view
to try to get some treatments that are actually effective. Right now, there's not much out there that has any significant impact in making Alzheimer's patients any better.
So they're definitely trying to look at not only the patient genetics, but also what's going on with the gut microbiome, who's there, and
What are they doing? And then, of course, they're trying, it's always challenging unless you do longitudinal studies. Again, like, well, is just because they get Alzheimer's the reason their microbiome is different? Or does the microbiome change and that leads to Alzheimer's? So there's, kind of like the chicken before the egg type idea. And so that can be tricky, but they are definitely looking into this as a potential angle to pursue. And some studies have shown some very interesting data implicating that with a dysbiosis of the microbiome in the gut, it can be showing up more commonly in Alzheimer's patients. Okay, so this whole idea of what are they doing in the gut that can be influencing the brain, they know it's really firmly established that microbes in the gut can produce immune active and neuroactive substances. So this is a partial list of what I found in the literature. Gamma aminobutyric acid, often called GABA, melatonin, dopamine, serotonin, acetylcholine, histamine, catecholamines. So if you've taken any neuroscience classes or physiology, perhaps this sounds a little familiar. So they can see that, the precursors to these final products, they can go into the bloodstream and can get across the blood-brain barrier and then potentially influence what's happening with your brain and neurotransmission. So this next image is actually taken from your book in chapter 14. They're just trying to show the idea here that you have microbes acting on, creating these molecules. And so in this case, they're just showing GABA and histamine. And so there's a lot of study looking at what are they producing? Is it getting to through the blood-brain barrier? And then, of course, the long-term idea is, can we create probiotics that are specifically designed to ramp up neurotransmission of some sort, depending on what's going on with the patient. So I like this quote: as much as a third of an animal's metabolome. So all the stuff that's showing up in your blood when you get your blood taken, not just red blood cells that are showing up there. You have a whole suite of chemicals. As much as a third are coming from the microbiome in your gut. Okay, the circulatory system extends the chemical impact of the microbiota throughout the human body, transporting metabolites that influence the physiology and metabolism of distant organisms, and potentially influencing other bacteria elsewhere on or in your body. So if you can imagine, of course, your blood vessels go through all of your whole body. And so that's just like a super highway for these microbial products to course throughout your whole body. And that's why they're saying, of course, it can influence distant parts of your body. So there's a example, one of the earlier studies was showing this Mycobacterium vaccae. This is not dangerous. It's actually a normal soil microbe. And what they were finding is that people exposed to it had decreased stress levels. And because it is increasing the serotonin in the person. And they did this with mice initially, and they were finding that I'm not a mouse experimenter, but they have they agree that there's certain tests you can run the mouse through to demonstrate whether they're stressed or not.
like a maze test, give them a novel environment and see how stressed out they get by it. The forced swim test, I think that's when they put them in that big round vat of liquid and, they make you swim and swim and swim, and then they can test for stress, chemicals from that. Coping strategy. So this one, I think what they did is that they gave, a better coping strategy when mice were confronted with an aggressive animal. I think they made the mouse get confronted by a cat. I think that's what they did. So I would have liked to have seen video of that one. So, but they said it helped the mouse deal with that situation. They've looked at germ-free mice and said, what happens with when they don't have any microbes in their gut, what's going on, and they could see that they definitely did have altered brain chemistry, which led to anxiety and depression. And apparently there are like tests you run the mice through to determine if they're anxious or even depressed. So like I said, I'm not a mouse experimenter, but they have like apparently good studies to demonstrate that. So, and they also see that this gut brain connection is faulty. It's not normal if you have germ-free mice. And so the idea that having a good, diverse, high populations of the microbiome in your gut can actually help you think better, remember better, reduce anxiety and depression, that is a very tantalizing idea for personalized medicine. Like, if we can better understand this, especially patient by patient, then we could really target and give them the proper needed microbes in order to support better health or better brain functioning. So they're really, when this one came out, this was in 2018, this was really one of the major papers at that time that was finally saying, we need to think about the microbiome. And so it was an exciting time during all this microbiome research. So some scientists, when they published their data, they actually were able to show that you can transfer microbes and get the transfer of depression. So again, showing this idea that if you take feces from a depressed patient and put it into a germ-free mouse, it would show the hallmarks of depression in the mouse model. Like I said, they have apparently ways to determine that. I don't know all the details, but yeah, transferring the blues. So when that got published, this was in 2016, that was a big wow moment because they actually did fecal microbiome transplantation from the patients and then put them into the germ-free rat model. And we're able to show that the rats got depressed and their microbiome level, their population and diversity went down. So they're saying, just doing that was able to show in the rat model that you could transfer those qualities of depression into the rat. So that's pretty strongly implicating the microbiome is involved in this process of depression. So that was some of the earlier research. And of course, they made the point in that paper that if we could just better understand this, then we could do personalized medicine with certain probiotics and prebiotics to help with this. So other papers have come out since then. And they're targeting inflammation, which we've been talking about, this idea of internal systemic chronic inflammation can lead to all kinds of health implications, in the sense of obesity or heart issues, but they're also saying even mental function. And so depression and anxiety and things like that. And so they're really trying to look at this, what's going on with the microbiome, is inflammation involved? And it seems to be yes and yes. They are showing that both of these issues are playing a role in mood. And so here, this was the abstract from that 2019 paper. And They were using germ-free mouse models again. And they were looking at what's going on with the immune system and the brain. Gut microbiome is playing a facilitating role between stress response, inflammation, depression, and anxiety. And so they're starting to collect enough data that they're able to feel more confident that the microbiome and inflammation is causing, how well your brain functions. So this is one of the images from the paper where they're trying to show visually what's going on here. And so this is if you have a stressful situation. So they're saying it can work both ways. If you feel stressed out, you produce cortisol that can alter your gut microbiome. It can lead to metabolites getting through the endotoxin, that's the gram-negative membrane, the lipid A in the membrane of the gram-negatives. And so they're saying how all of that can lead to inflammation. And then they showed here, they're saying, okay, what if, and you have a person that's stressed out and we try to do some things. So here they're saying, if you have microbes in the gut that are producing, we'll talk about short chain fatty acids, that's considered really good. You want microbes doing that in your gut. And they're saying, if you have the right products being produced by the microbes, you get a good intact intestinal lining, good mucus lining, your endotoxin levels go down, so you don't have as much of that in your blood, which means your inflammation levels go down. And so they're really looking at this idea of stress and inflammation. And it can work both ways, just you being under stress can alter your microbiome. So again, this is a 2021 paper where they're looking at the same idea of anxiety and depression. And they're linking it again to inflammation. They're saying if people are under anxiety and depression, you see a higher abundance of pro-inflammatory species. And so, and then lower short chain fatty acid production. And so again, they're piecing it together that if you can lower inflammation, chronic inflammation, if you can get a good mixture of microbes that can produce lots of short chain fatty acids, that might really help. And so, and it's a heck of a lot cheaper taking a probiotic than some of the medications that they prescribe. Fermented foods, that's one of the reasons why I wanted you to do a fermented food extra credit, is with this in mind, that it can really improve not only your physical health, but I've read enough papers where I think they're onto something with the mental health idea. I know I feel mentally much better when I incorporate a lot of fermented foods myself, but of course I'm one, and a sample size of one, but I seem to notice a difference if I ramp up my fermentation foods. But that's something pretty simple, right? I mean, that's something we can all do. Hopefully, a lot of you took advantage of the option to try fermenting your own foods. It's not that hard. Can be fun and tasty most of the time.
You can still do that, right? That's the thing.
Oh yeah, you can still do that. By the way, water keeper grains I ordered a long time ago and they lost it. The United States Postal Service lost it. So I ordered again. And they claim it's coming in next week, which isn't giving me much time, but as soon as I find that it's delivered, I will give it to Duncan and Isabel, but frustrating. Unfortunately, I don't know of any place that does water keeper in town that would sell it, or, I mean, the grades. Okay, so they're really looking at this carefully. I think it's a great idea, looking at the idea of something simple that we can all do. So this gets into the psychobiotic term. This is a new term, relatively new, certainly newer than the probiotic term. So enough data has come out where they're realizing key microbes can be very important in influencing your brain function. And so they're
Calling it a separate category. They're saying they're psychobiotics. So technically that's probably what you would ask for from the doctor if, you know, in the future with personalized medicine. And so what they're looking at here is, can we find microbes that either make neurotransmitters or precursors to that? And so, you know, again, just looking at some of the things that they're studying. Brain-derived neurotrophic factors is a new one I didn't put on the previous list, BDNF, but like again, I'm not a neuroscientist, but Dr. Adams said BDNF is like miracle grow for the brain. And me being a botanist, it's like, oh, I get that. Because miracle grow is really good for plants, helps them grow and serves as a fertilizer. So I thought that was a good analogy. But so they're looking at designing these psychobiotics so that you can get personalized medicine prescribed to you. They're saying, you know, in the future, you should be able to control mood, cognitive ability, learning, and memory, which would be awesome when you're going to college to be able to improve your learning and memory. So anyways, I think a lot of this can happen already just by eating fermented foods and trying to make sure that your microbiome is fed correctly. I think can go a long way to making you feel better. So all of these terms, psychobiotics, probiotics, you know, you start looking at this, and so the probiotic industry has just skyrocketed. There's all kinds of probiotics out there, so be really careful. But they said, you know, if you're going to build one and sell it, it should be microbes that hang out with humans, not some exotic microbe. They shouldn't have any virulence genes, so they have to do the DNA sequencing. They should be able to get killed off by antibiotics. You want them sensitive. You don't want them resistant. You want them to be able to make it through the stomach. Catalase negative. When that was on the list, I'm like, I had to think through that one. Like, catalase negative, what's the big deal there? So catalase negative, if you're catalase positive, do you remember in lab, you took microbes, put it on a slide, and then do you remember what you dropped on it and looked for bubbles? Hydrogen peroxide. So you were looking for oxygen, you know, coming off. So basically what they're getting to here is you don't want aerobic bacteria. That's basically what they're saying. I don't know why they didn't just say that, but so they're saying, yeah, catalase negative would be good. You want them to be able to persist. You don't want them to wash out. So if they could attach to the intestinal mucus, that'd probably be the best. And able to stand their ground, so to speak, with the microbes that are currently in your gut. They don't, you know, again, you want them to persist. You want them to produce short chain fatty acids. I'll get more into that. And if you look on bottles of probiotics, the ingredients, almost every strain is a gram positive. At least current probiotics I've looked at on the shelf, they're all gram positive. And that is because if you get too many gram negatives in you, that's the LPS, the lipopolysaccharide, the gram-negative cell wall that has that lipid A that's an endotoxin. Too much of that in you is not good. Okay. All right. I guess we got to stop. Thank you for your patience. We'll continue on next time.